In this video, we are going to demonstrate an epigenetic phenomenon known as genomic imprinting. Genomic imprinting creates a complex inheritance pattern that can be observed in families with certain genetic conditions. In our demonstration, we will use candles to represent a gene or a set of genes. When a candle is on, that means the gene or set of genes is expressed or actively being used. For the most part, we will show candles in pairs like this because there are two copies of almost every gene in your body. One of these copies is from your mother, and one of these copies is from your father. For many of the genes in your body, both genes are equally useful and are necessary for you to be healthy. Both genes are expressed, or turned on, to produce protein products that are used by our cells. This is our example family. The top row has 4 grandparents, the middle row has 2 parents, the children of each grandparent pair, and the bottom row has the parent's child. Blue squares represent men, pink circles represent women, and the purple diamond represents a child whose gender isn't important for this discussion. We are going to place candles on each symbol to represent a gene or set of genes of interest. First, we will demonstrate non-imprinted inheritance. You will notice that each person will have 2 candles that are both lit to show that it is necessary to express both gene copies. The grandparents each pass 1 copy of their genes onto their kids in the second generation. Both kids, the parents, will have 2 candles that are also both lit up. And finally, when the parents each pass 1 copy of their genes onto their child, both the child's candles will also be lit up. This is how it works for many genes, but for some genes, this process is more complicated. Even though you still get 2 copies of the gene, one from each of your parents, only 1 copy will be expressed, and it will only be expressed if it is from your mother or, vice versa, from your father. This is known as genomic imprinting, where 1 copy of a gene, depending on parental origin, will not be expressed and will be silenced or turned off. These genes are silenced through an epigenetic modification called DNA methylation, which does not change the DNA sequence, but does change whether it is expressed. For paternally imprinted genes, the expressed gene is passed down through the mother. For maternally imprinted genes, the expressed gene is passed down through the father. Genetic complications can arise when a child inherits an expressed gene that does not work properly. Let's explore this further. Remember, a candle that is lit means the gene or set of genes is expressed, and when it's turned off, the gene or set of genes is methylated and therefore silenced. To understand how imprinting happens, you must imagine what is going on in the egg or sperm during gametogenesis, the process that creates these cells. When we make sperm or eggs, they receive only half of the total DNA that the rest of our cells contain. This is because when a sperm eventually fertilizes an egg, their DNA will combine to form a new human with a complete set of DNA. So, only one of the parent's 2 gene copies, only 1 candle, will go into a given egg or sperm. With non-imprinted genes, both copies will be expressed, so each sperm or egg will contain an expressed gene, a lit candle, that will be inherited by the child. With imprinted genes, this is a little more complicated. Most of the cells in parents with the typical expression profile for an imprinted gene will have 1 gene copy that is expressed and 1 that is not. But because imprinted genes are expressed or not based on which parent they are coming from, the methylation patterns must be reset in sperm and eggs to be consistent with either paternal imprinting or maternal imprinting for the next generation. When a gene is paternally imprinted, that means any copy of the gene that comes from the father will be silenced. Half of his sperm receive the expressed copy and half do not. As a result, the methylation pattern is reset in the sperm that do in order to silence the previously expressed gene. This means the father will only produce sperm that have silent gene copies. So, even though he himself is expressing 1 gene, the child will only inherit a silenced gene from their father. That child then relies on their maternal gene to be expressed. Like the father, the mother also only expresses 1 gene. Half of her eggs receive the expressed copy and half do not. The methylation pattern is reset in the eggs that don't in order for the gene to be expressed. The child will therefore inherit an expressed gene from their mother. This is paternal imprinting. Maternal imprinting works the same way, but instead the maternal genes are silenced and the paternal genes are expressed. Please note that even though which parent the gene is coming from matters, it doesn't matter whether the child is a boy or a girl. Now that we know how imprinting is inherited, let us revisit our family. Let's demonstrate inheritance for a paternally imprinted gene. Remember, in paternal imprinting, the paternal gene copies that are passed on are always silenced and the maternal copies are always expressed. Again, note that it doesn't matter precisely which gene copy the next generation receives from their parent. The fathers must be silenced and the mothers must be expressed. We can also demonstrate inheritance for a maternally imprinted gene. In this case, the maternal gene copies that are passed on are always silenced, and the paternal copies are always expressed. Now let's use this model to show how inheritance for a specific genetic condition works. We will use the examples of Prader-Willi and Angelman syndromes. The genetic variants that occur in Prader-Willi and Angelman actually occur in the same region of DNA, but different parts of this region are paternally or maternally imprinted. Let's start with Prader-Willi. Prader-Willi syndrome is a maternally imprinted condition caused when the DNA passed on by the father is missing information, rendering it non-functional. Here, we are showing the non-functional genetic region in green. You will notice that the father's functional copy of this region is expressed and his non-functional copy is silent. Therefore, we can tell that he inherited the non-functional copy from his mother and it has been imprinted. Half of his sperm received the functional copy and half received the non-functional copy. The methylation patterns are reset so that they all will be expressed. And when the mother produces eggs, all of her gene copies will be methylated and therefore silenced. So we see that the child inherits one functional copy and one non-functional copy, and due to the maternal imprinting, only the non-functional copy is expressed. Because the functional copy is imprinted and therefore silenced, it is unable to rescue the paternal gene dysfunction. This causes Prader-Willi syndrome. Here is an example of how Prader-Willi can be passed along in a family. It is only seen in the child because of the way the imprinting has unmasked the non-functional gene region. In the case of Angelman syndrome, where the genetic region that's missing information is shown by an orange candle, a mother can pass it on because it is paternally imprinted. Therefore, the methylation will be reset in all her eggs to express both of her copies. At the same time, when the father produces sperm, all of his gene copies will be methylated and therefore silenced. That way, the child can inherit a non-functional copy from the mother and a functional one from the father. And because the father's has been silenced due to paternal imprinting, only the non-functional one will be expressed, causing Angelman syndrome. Here is an example of how Angelman can be passed along in a family. Just as with Prader-Willi, it is only seen in the child because of the way imprinting has unmasked the non-functional genetic region. The take-home point to remember about imprinted genetic conditions is that there needs to be a change on the non-imprinted gene copy that impacts its functionality, and that imprinted inheritance can either mask or unmask this change. That's why some people can have the change but not the condition, and others have both. Thank you for watching this demonstration video. We hope this helps to illustrate how imprinting works and how imprinted conditions are inherited in families.